Hello everyone, my name is Neda Barjesteh. I'm an assistant professor at the Faculty of Veterinary Medicine, University of Montreal. First, I would like to thank the Canadian Poultry Science Network for providing this great platform. Also, I am delightful to be here and present results of research project conducting in my lab since I joined the Faculty of Veterinary Medicine in 2018. The overall objectives of a research program in my lab is to determine the underlying mechanisms involved in the initiation and regulation of innate antiviral responses against viral respiratory infection in chickens, with the main focus on avian influenza virus and infectious bronchitis virus. As you would know that there is a constant battle between virus and the host. Virus infects, replicates, and spread to survive. On the other hand, host cells can sense the presence of viral component and initiate immune responses to limit the replication of the virus. Therefore, the interaction between the host and pathogen, also factors related to host, pathogen, and environment determine the outcome of infection, which can lead to a disease. But the one thing that I would like to highlight here, that communication between different host cells is really important, and also communication of cells of the innate immunity is important, because this communication can shape the adaptive immune responses as well. Therefore, in a research program in my lab fall into two main categories. First, basic and discovery research. On the other hand, applied research. With respect to basic and discovery research, I'm interested to determine the antiviral responses, specifically in epithelial cells, because I believe that they play a critical role in the initiation of antiviral responses, because they are the main target of several viral respiratory infections. Also, I'm interested to determine the possible role of extracellular vesicles in the in antiviral responses. And as we would know that following the infection of host cells or activation of host cells, there are several intracellular signaling pathways, and the regulation of these signaling pathways is really important. I'm interested to determine possible role of microRNA in the regulation of antiviral responses. With respect to applied research in my lab, we are interested to determine how we can use microRNA for vaccine development. We are looking for the alternative approaches for control of viral infections in chickens. Currently, we are we are trying to find a candidate microRNA can be used as immunostimulatory molecules or vaccine adjuvants. Also, because my lab is set up to isolate, purify, and produce extracellular vesicles, we are interested to use extracellular vesicles as a delivery system. We can use them as the immunostimulatory molecules to modulate antiviral responses in chickens. As I mentioned, epithelial cells are the primary target of several viral respiratory infection, and following the infection or stimulation, they release chemokines, cytokines, to communicate with other host cells, including macrophages or dendritic cells. Also, now it has been shown that EVs, extracellular vesicles, release from epithelial cells, and they can play a role in cell-to-cell communication. You may ask, what are these extracellular vesicles? We have different type of extracellular vesicles. Most host cells can release extracellular vesicles, and depending on their size and the biogenesis pathway, we have macrovesicles, we have exosomes or multivesicular bodies, and we call all of them as extracellular vesicles. And as I mentioned, extracellular vesicles released by host cells. These extracellular vesicles contain several bioactive molecules, including proteins, RNAs, and even they can carry a viral component to activate other cells like macrophages. So in my lab, we want to determine possible role of extracellular vesicles. In the first step, we wanted to profile their content with respect to microRNA and proteins. Also, we wanted to see how these extracellular vesicles can impact the immune system. Therefore, first we isolate and purify the EVs released from different chicken cells, including tracheal cells and macrophages. And after that, we wanted to determine their content. Therefore, we prepared tracheal cells by tracheal organ culture. We infect them with influenza virus. We infected them with influenza virus or other stimuli like TLR ligands. And following several steps, we isolated EVs and characterized their profile. My PhD student Kelsey will present the proteomic study related to this project, and here I'm going to present the results related to microRNA. And what we found that EVs, they carry microRNA, but the profile of microRNA can depend on the stimuli. And as you will see here, each treatment, we have different treatment here, and we see that the profile of microRNA depends on the type of a stimuli. Also, the profile of the microRNA profile can be different between cells and EVs. Therefore, there was a question if these microRNA or other proteins can randomly shuttle into be in the EVs, or there is a specific process can shuttle microRNA into EVs. And previous study highlighted the possible role of RNA binding protein in this process, and they can selectively shuttle microRNA into EVs. And because we know that microRNA can affect affect the signaling pathways, we wanted to see the potential target genes of our microRNA, and we determined these potential targets. Mostly, we focus on pathways involved in antiviral responses. Also, it was interesting for us to determine if this microRNA can target viral genome, and we showed that differentially expressed microRNA in our study were able to potentially target multiple segments of the AIV viral genome. And because we want to use this microRNA in vivo, to use them in chickens, now we are trying to find our candidate microRNA. We have a criteria for this selection. For example, expression profile in different treatment groups, predicted targets, as well as reference to literature. Based on this criteria, now we selected several microRNA. We are focusing on the function of this microRNA, and here I'm just presenting the preliminary result that we have. We determined the direct effect of microRNA, this microRNA LET7F on chicken macrophages, and we showed that these first, we can transfect cells with these microRNA using different dosage without having any cytotoxicity effect. They can activate macrophages, and also because they can activate these microRNA can with LPS can activate antiviral responses. It's possible with this treatment we can interfere with the replication of AIV in chicken macrophages. After that, we ask a question: do EVs secreted from chicken tracheal cells have an impact on other cells like macrophages? Because we know that macrophages have several functions, including nitroxide production, phagocytosis, and induction of innate responses. Therefore, we wanted to see that if macrophages are able to uptake EV, or they can just sense EVs. We had a simple experiment to show if macrophages can uptake EVs. Sorry, the fluorescent picture may not be clear here, but by using different fluorescent dye, we have been able to show that actually macrophages can uptake EV, as you would see here, comparing to our control groups. And then these EVs are able to activate macrophages because we look for the phagocytosis and nitroxide production. Also, we found a kind of synergistic effect of our EV treatment with LPS treatment, because we know that LPS is really good TLR ligand to activate macrophages when we compare them with control group. Also, we check for the induction of the expression of some candidate genes involved in pro-inflammatory responses and antiviral responses. Just giving you an examples here, we look at the expression of IL-1 beta and PKR as interferon assimilated genes, and we showed that by using even low concentration of EV, we are able to induce the expression of genes in chicken macrophages. That brings us to the end of presentation. I would like to summarize by saying that chicken tracheal cells are able to secrete EVs, but the content of EVs can be influenced by the type of stimuli or infections. They are enriched in bioactive molecules which are involved in immune responses and cell signalings. They are able to impact the function of other cells, including macrophages. We are still trying to determine the underlying mechanism in this cell-to-cell communication. And thank you so much for your interest and attention to this presentation. I would be interested to hear from you with your thoughts and questions. Thank you.
Thank you very much, Dr. Barjesteh, for your presentation. It's, I'd say, for a young professor, putting together such a comprehensive research program is quite a task. Congratulations.
Thank you. Thank you so much, Dr. Boulian.
I have, unfortunately, no question from the audience, but I do have one for you, though.
Sure.
Oftentimes, I guess that the industry is looking at research like yours that is more fundamental but essential to further our understanding of diseases in poultry. But how do you foresee the development of your research and the possible application to the field?
Yes, exactly. As I mentioned, thank you so much for your question, because I'm thinking that it's really important that we know that at the end of the day, we can have a kind of applied research and employ them for industry, because whatever that we are doing in academia, we want to employ them in the industry and be beneficial for the industry. So for me, as I mentioned, and I'm focusing on microRNA and EVs, and I want to use them, for example, EVs as a kind of delivery system that if we want to, for immunostimulatory molecules to use in the poultry farms, or for vaccine adjuvants, we want to have a delivery system. So whatever that I'm doing, I'm trying to develop a system to be used in chickens. And with respect to microRNA, what I'm doing, I'm hoping to find candidate microRNA that we will be able to use them in the poultry industry as immunostimulatory molecules, as adjuvant molecules. So because we know that they have a role for the tailoring immune responses. So this is just a matter of time that I'll be able to find our candidate microRNA to be used in the poultry, because they are safe. And I want to mention that they are safe and we can use them. And the only thing is that now I'm working, and also they should be cheap for the industry. So.
Well, that's the ultimate argument for as a selling point.
Exactly.
There's a question from the audience. It is the following. Do you also have plans to incorporate other immune cells and assess their role in stimulating EV's production by epithelial cells? Is this possible in vitro?
So sorry, I'm trying to understand. Yeah, the thing is, in my lab currently, I'm working on the EVs released from other cells. For example, as I mentioned, I'm working with macrophages. I want to see how about EVs released from macrophages. The one thing that I want to do, I want to see how they impact on the other cells. But the only thing I don't understand,
cells like heterophils, for example, are you planning? You're targeting macrophages, but is it a possibility to target other immune cells such as heterophils or
exactly, because mostly first I'm looking for the pathogenesis of the disease, which type of cells involved in the induction of immune responses. So if we imagine that the respiratory system infected with several viral infections, we have macrophages, we have dendritic cells. After that, even we have, as you mentioned, heterophils, or then we have T cells coming toward the tissue. So now I have a plan to not only for working on the macrophages, also the one thing that currently we are doing, we want to see the effect of EVs released from epithelial cells on the function of T cells. So it is a kind of bridge between innate immune responses and adaptive immune responses.
Oh, well, you'll be busy for the years to come. Well, thank you very much for your presentation.
Thank you.